the transcriptional activation or repression is not established the way it is being presented in the textbooks. I can give you one example. I mentioned about H3 36 methylation, right? What is this mark? Does anybody know?
Sir, it's an activating methylation, I guess.
Yeah, so that's what is being taught, right? So you have histone modifications which are activating another inactivating, and then you have reader enzymes and writer enzymes and so on, right? So I hope you understand the reader writer thing, right? Reader writer erases. Histone deacetylase is a eraser enzyme, right? Histone methyltransferase are writer enzymes. Reader enzymes are your chromatin remodeling complexes. So this is, as somebody said, this is activating mark. Well, this marks the gene body, right? This mark is present in the gene body, right? And particularly the gene body of transcribing gene. You might say, how is that different from others? Well, other marks are present at two other sites, not this site. They are made too distinct. This is actually not that far away. This is coming at proximal site here. So most of the histone modifications are at PSS proximal sites, right? Let's say histone acetylation, S3K9 acetylation, S3K14 acetylation, S3K4 trimethylation, S3K4 monomethylation, and so on. They are coming approximately. So this is that. So if you find this mark in the gene body, it means that gene is transcribing. So if you go by the histone code hypothesis or whatever we discussed 2 minutes ago, it would tell you that this mark is present, then the transcription happens, right? The full length transcription from PSS start site to transcription termination site is happening because this mark is present. That's what you would assume if you follow the histone code hypothesis. In fact, I can tell you that for this mark, it is established already that this is not the cause, it is the consequence. I'm sorry if this is being repeated, somebody had already taught you. But transcription is not proceeding as CK36 primethylation. It is preceding CK36 primethylation. Right? Transcription happens first, and then you have DNA open up for transcription and all that to protect that DNA from mutations and to attract the DNA repair machine ESTK56 marks is deposited out. So it has been shown at least in few model systems. Likewise, you have another mark which is gene body mark for the transcribing gene, which is S3K79. So this is not well established. Cause and effect, whether most of the chromatin modifications are the cause of transcription activation and repression, or whether they are the consequence of it. It's not well established. And there is a controversial paper from a few years ago where DNA methylation was shown to be not shown to be causally linked with transcription repression. It in fact is a consequence, not the causing mechanism. So although majority of the papers or articles studies, they do claim that there is a causal relationship for most of the marks. There are evidence out there which suggests that this is not yet done, this is not fixed. Okay? People are still working on it to really establish what is following what. Nevertheless, when we use the word mark, right, chromatin mark, histone mark, or modification, that notional thing is correct. We already know that there are marks which are associated with transcribing genes and the there are marks which are associated with repressed genes, right? So that much clarity is out there. So established. And that is important to really look at the whole genome by dividing the genome into different unique entities. Right. Okay, so this was just one thing that I wanted to highlight today. The other thing is, let's look at various kind of marks, various kind of genomic elements out there. So let's say you have promoters, right? What histone modifications? Now, how would you identify if a given element is a promoter? What can you do experimentally, computationally, or by any means, which will suggest you that this is promoter?
CPG islands should be present over there, I guess.
CPG island. CPG islands are actually present more on the. If you compare the expected value, the observed value versus expected value of CPG content, which is C followed by G, right? Number of times C follows G's, and the promoter region divided by number of C's times number of G's. Right? That's the expected value. And if you look at this value, it is actually depleted in the housekeeping genes. Most of the actively transcribing genes are actually having lower than expected CPG content. So whatever you said might be true for the genes which are tissue specific, not the housekeeping. Sorry, this yeah. So housekeeping ones are having CPG. That's true. So you will have promoters will have whatever I said the other way around. The housekeeping genes will have more than the expected CPG. So that's one thing. So if you plot the CPG content, I think this was taught in the report. So we did an exercise on that, right? So if you plot the CPG, that's correct. Okay, so you will have some peak out there. Fine enough. So let's talk about in the context of the present course. What else can be done to identify whether this is promoter or some other element?
Presence of Tata box.
Tata box, there are genes which are Tata-less genes. Okay. So that's not there is a Tata linked genes and there are Tata-less genes out there. Tata box containing genes, Tata box non-Tata box genes. I repeat that in the context of present course, what will you look at? Will you look at some histone modifications? Acetylation. Somebody said acetylation, right? Then of course still start codon. Okay, fine enough. People are going back to basic biology. Somebody said histone acetylation, right? Histone acetylation. Alright, histone acetylation, but that's also present on the enhancers. If nobody taught you, then I'm giving you this information that it's also present on the enhancer. Fine enough. What else? So histone acetylation, that's the correct answer, but I can tell you that this is not sufficient. S3K4 acetylation, S3K9 acetylation, S3K14 acetylation, and so on. There are more acetylations. What other marks would be present at promoters? Well, I just spoke about that in the previous slide here, right? I mentioned that you will have S3K4 Me3 prominently present at promoters. Okay, so this is promoter associated mark. However, whatever we wrote over here are also present on an enhancer. Me1. And then if you go to transcription factor binding site, you will have Pol2 enrichment and other transcription factors binding things and all that. But let's say we only talk about a stone modification. So then the problem is still there, right? How do we identify, how do we distinguish promoters from enhancers? We have all kind of, let's think of a scenario, not a scenario, it's a reality now. We have all kind of stone modifications. Most of the stone modifications which are known in literature are mapped across different cell types, tissues, organisms. Right? You have that data. Now I tell you to do a small exercise to tell me where all the promoters are without looking at the annotations and where all the enhancers are. See, if you look at the annotation, you can be misled. The reason is following. There will be regions which are unannotated. Okay, then you would say that these are all enhancers. Well, it turns out that they were proximal to the promoters, and those were unannotated genes, unidentified genes. What do you think? Where would be the difference? Would there be any difference between these marks for enhancer and promoter? All of them are present at both the sites. These are present at the regulatory elements. Activating regulatory elements, they are present on those. Well, what I could tell you, a very easy thing today is that there is a difference out there. One difference which is easy to understand is that you have more of, so you have for promoters, so let's say this is your promoter over here. That's your promoter, right? At promoter, you have S3, K4, trimethylation, very high. Okay, so if you look at the peak, so the peak would be very sharp at this location. And the same peak, let's say you have enhancer over here. This is your enhancer. The same peak of S3K4 trimethylation would be present certainly on enhancer also, but peak strength would be lower. Okay. Then if you look at another mark, which is S3K4 ME1, just think about it. ME1, the presence of ME3 would actually depend on presence of ME1, right? Still, this is a very reliable mark. So if you look at ME1, ME1 would be high at enhancer and lower at promoter, which means that most of the ME1 marks are converted to ME3 by sequential methylation, right? Monomethylation, dimethylation, trimethylation. So it is already converted to trimethylation, but that doesn't happen significantly at the enhancers. Okay. So now one of the students said that you can look at the downstream transcription start site, that will be a promoter. Another way, this can also be extrapolated to another thing, that look at the transcribing region downstream, right? If that is, there is some level of transcription downstream to the regulatory element, that is likely to be promoter. And if it is not, it is enhancer. That's also incorrect. The reason is following, both of them transcribe. So you know that the promoter region transcribes the downstream gene. So this is going to transcribe this thing, right? However, this might be new to you people that enhancer also have transcription. That's called eRNAs, enhancer associated RNAs. So what you're learning now is that the distinction between enhancers and promoters is blurring up. Okay, they have similar marks. Both of them will be enriched with acetylation and methylation marks, both of them are being transcribed. So then what is so special about promoter and enhancer? So we shouldn't look into regulatory elements in that way, that this is promoter, this is enhancer. They are regulatory elements, and most of the regulatory elements are having similar properties, at least the activating regulatory elements. Now, since I mentioned that people did find some difference between the enrichment of S3K4 trimethylation and monomethylation, what you could do is you can plot to identify enhancers from promoters or promoters from enhancers. You can take all the genomic elements that you're looking at in the whole genome and plot the ratio of S3K4 ME3 by ME1, maybe at log scale. Okay, and then plot the numbers here, number of those regulatory elements. This is the distribution plot. What happens is that there will be some value over here which is 0 because this is log scale, and you're going to get a distribution which is bimodal. Right, so these are your promoters and these are your enhancers. You have here demarcation of enhancer promoters by looking at S3K4 trimethylation and monomethylation. Why nature has chosen so, we don't know, right? But this is one. So as I said, we have to take histone modification as a mark for the time being. Okay, the moment somebody starts claiming the cause and effect thing, one has to come up with a very clear evidence for the same. Okay, now there is a bizarre consequence of this. Okay, the moment you have this kind of thing, what if there is a promoter which is actually coming here? So let's say there is a promoter coming here. Somewhere here there is a promoter. You already know from literature and from your own experiment or information that this is actually promoter, but this is this a false positive as an answer? This should actually come over here, but it's coming here. So what's happening? So I can actually show you the data. Yeah, so here is the thing. If you see the slide here, on the left is S3K4 trimethylation, on the right is the monomethylation. And what we are plotting is each row is one element, okay, one region of the genome, and what we are plotting is the following. This is basically right in the center is your transcription start site, and each row is a promoter here. Okay, so each row here, each row that you see here is a promoter, one promoter, annotated one. Okay, promoters. And the center here is your transcription start site. And what you are plotting is the S3K4 trimethylation with respect to transcription start site of all the promoters, right? So this is basically you take all the transcription start site, and this is for gene 1, gene 2, gene 3, all the start sites center and just align them. Okay, this is the start site, and make an aggregation plot. Okay, and this is the aggregation plot that I'm going to make now. What you plot is with respect to distance from transcription start site, you take let's say minus 5 kb here and plus 5 kb here. Okay, and plot the enrichment of S3K4 trimethylation, which is coming from a ChIP-seq experiment, and then you will see some kind of enrichment like that. And that's why you have red color in between, because there's a higher enrichment, it's a heat map. Likewise, you do the same thing for the S3K4 monomethylation. This is your TSS site in the center, and then you see that there is a bit broader distribution over here, but nevertheless, they're centered around the transcription start site. Now, once you cluster them, so you see that similar things are clustered over here. These are the ones which are having very low signal, right, in both the cases. What you see here is that in this red color here, what we are trying to show here is these are typical promoters. Right? Promoters. It is having high S3K4 trimethylation, right? Me3 is high and Me1 is low. Okay, here you have some kind of heterogeneous mixture, could be because the neighboring region is having some enhancers. This is some kind of heterogeneous thing. So let's not talk about this for the time being. What is striking is this part. What you have here is there are promoters which are higher in Me1, and lower in Me3, right? These are promoters, we know that from literature, these are promoters, but they are having marks which are resembling enhancer. So are these false positive coming as enhancer, but they were actually promoters? No, in fact, what happens is that these are known as enhancer-like promoters. Enhancer-like promoters, okay, or sometimes they are known as E promoters. There are more detailed analysis now being done on these promoters. So what's happening is that there are promoters in the genome which have an enhancer potential. No, somebody is asking, is this the only way to distinguish enhancer and a promoter? No. I'm telling something in the context of this course. Okay, if you really want to get into the genetics of this, perhaps genome structure and function course would suit better, or maybe at the first year, second year courses. The other differences you already know. One of the striking difference that you can think of is the orientation. Okay, promoters have directionality. Okay, there is a direction of the promoter. Okay, if you reverse it, the transcription happens on the other side, right? So there is a directionality, orientation of promoters, but enhancers have no orientation. Okay, that's the only, that's one of the difference. The other difference is the most of the enhancers will be surrounded by more number of transcription factor binding sites as compared to enhancers. The enrichment of transcription factor binding site TFBS will be very, very high. Let us say I am plotting it here, transcription factor binding site density. Then that density would be lower here, but very, very high here. TF density. Okay, there could be these kind of differences, but I'm talking in the context of the present course, in the context of epigenetics, what are the differences? Okay, and relatively, acetylation marks are generally kind of equal, but if you want a detailed combination of acetylation and methylation marks, that kind of analysis I think has been done, and people find a peculiar combination of acetylation and monomethylation which marks the enhancers. But for the simplicity, for the explanation, I took methylation as an example, which nicely distinguish enhancers from promoters. What I was coming down to now is that what happens to the promoters which have enhancer-like marks? Do they have enhancer potential also? The answer is yes. As of now, from the episomal system, if you have a synthetic system to check the enhancer activity, let's say this is your luciferase gene in an episomal system, and at the enhancer location, you put the promoter, typical promoter. Okay, this is your, let's say, TP, typical promoter, and look at the downstream transcription through this promoter, through this enhancer. Okay, this is enhancer position, so let us write it enhancer position, not the promoter position. So there is a low transcription. However, if the same gene, same luciferase gene or any reporter gene is actually under the influence of an enhancer promoter, EP. Okay, it turns out that now this gene transcribes significantly. So this is having enhancer potential. Although this inserted element is actually a promoter sequence. Okay, so and there are enhancer promoters out there in the genome, which really complicates the scenario. Now think about what is enhancer and what is promoter. Enhancer is enhancing the transcription, and now enhancing the transcription of a distant gene, right? Enhancer can enhance the transcription of a distant gene. Can promoter do this? No, promoter needs to be proximal to the its downstream gene, right? However, what we now learn is that there are promoters of one gene which are regulating the transcription of other gene. Okay, so what I'm trying to say is the following. You can have a scenario where there is a gene over here, and in between there are maybe two other genes. Okay, and then you have another gene, and you have this promoter of this gene is actually like an enhancer promoter, enhancer like promoter, while this gene's promoter is a typical promoter. Okay, now what's happening is that this enhancer promoter can actually influence this target promoter, okay, and transcribe that gene, and this is working like an enhancer, right? Like an enhancer. Now, how this happens in space, we will learn later in this course when we talk about higher order chromatic conformation. But this really brings the network notion in the gene regulation, physical gene regulation, that if you have all the promoters, look at which one is interacting with whom, okay? And then there is a one population of promoters which can actually trans-regulate the other promoters, which means right at the physical interactions of genomic elements, you can have transcriptional regulation. You don't need enhancers to be part of regulatory network. Of course, they are part of, they contribute significantly to transcription regulation, but the promoters themselves can trans-regulate each other. Okay. Now, okay, so this is one thing. Now let's talk about enhancers in little detail. Since I draw this figure over here, I recall something else. So about enhancers, what do you know about enhancers? Where are they located, first of all? Where are they located, enhancers? The correct answer of enhancers location is that anywhere in the genome. Okay. Of course, then there is a disparity in the distribution. What I meant by anywhere is that they can be quite closer to target genes, maybe within 10 kb. If you talk about human genome, could be 20 kb, 30 kb, 50 kb, maybe in a different chromosomal arm, or maybe on some other chromosome. Okay, there are sufficient evidence which suggests that the enhancers could regulate the target gene in trans from one chromosome to another chromosome. So theoretically, they can be anywhere, but more often they are within certain distance from the target gene. Generally, they regulate the nearest gene. Okay, if there is an enhancer somewhere, and you have a number of genes out there, and then on this side also, it will look for, generally more often they regulate the nearest gene. Okay, they may skip this one and actually regulate this one, just like this scenario, but you know the frequency of such events decreases over time. Okay, they can be anywhere. Now this has really misled people. This kind of thing, enhancer promoter thing, that people did think about. The problem with the science is that people start having assumptions about things, that enhancer is a non-coding element and blah blah blah. Enhancers can also be located in the exon of the gene. Okay, they can be located, of course, intronic regions are non-coding. I can tell you one example where people were misled for decades. Okay, so there is a gene called LIMB-R gene. So this gene was named as LIMB-R gene because malfunctioning of these gene, mutations in these genes, or there is a particular mutation in this gene, is actually associated with limb malformation. You can get polydactyly, right? You have extra finger of Hrithik Roshan, right? Some of these stars have multiple fingers. So that's called polydactyly. Or sometimes you have syndactyly, which is a condition like that, the middle two fingers get fused together, right? So these conditions are actually associated, people thought it is associated with LIMB-R gene, that's why they name it as LIMB-R gene. And there was a particular mutation in the intron of this gene, one of the intron, the first intron, there was a SNP, which is associated with, this SNP is associated with syndactyly or whatever, polydactyly or limb malformation. And for decades, people keep on thinking, well, we have annotated this gene. Okay, so this is associated with this diseases and this has this function. It turns out that this is nowhere related to limb malformation. Just look at how genome-wide association studies can be misleading just because the interpretation was not correct, just because we made assumptions. Okay, nobody tried to annotate what this SNP is doing. This is an intronic SNP, first of all. Okay, so it's not in the coding region, this is in the non-coding region. Then people thought, okay, this is working as could be affecting splicing or could be affecting the regulation of gene through mutation in enhancer region. So enhancer of the same gene located in the first intron, which happens, as this is not new. It turns out that there is a gene more than 1 MB away, which is well known for such effects, and that is SHS gene, which is sonic hedgehog gene. You would have known about this, you would have heard in many developmental biology course how it is involved in limb development and body plan and all that. It turns out that this gene this SNP is located in the enhancer of this gene. Okay, this is the enhancer. This is the enhancer over here, and the SNP was in the enhancer. So what was happening once you have this mutation in the population, transcription of this, if you have this mutation, then transcription of this was affected, and that was leading to limb malformation. So you now understand how complicated it is, right? Not straightforward, which the regulatory elements crosstalk with each other in a very complex manner. And each of these figures that I have drawn brings the network motion, right? How at the DNA level you have regulatory networks of regulatory elements. Okay, so that was one thing. Enhancers, promoters, right? Anything else that you have heard? Have you heard of silencers? Anyone? First of all, silencers are poorly understood. Okay, and there are reasons behind that. What is the reason? Silencers are not known or poorly known, or they are rather infrequent. Can anybody tell me? It's related to basic biology. How does repression happens? What is the most common mechanism of transcriptional repression? We're talking about promoter enhancer silencers because this section that I'm covering is leading to principal chromatin types. Enhancer is one principal chromatin type, which is combination of acetone acetylation and methylation, and I showed you the difference between promoter and enhancer. And later on, we will see that people have done multivariate analysis to actually come up with chromatin types, and we will see what are those chromatin types. But to understand that, I need to cover this fundamental. Silencers are not known or they are infrequent, and the reason is the mechanism of transcription repression is indirect. What happens is that you have, let's say, you have a gene and you have a activator. This is the activator. The the most common mechanism of repression is that repression comes and binds to activator and blocks, and then this cannot go to the target promoter and transcribe the gene, right? So there is no transcription. So this is indirect. The way repressor works is indirect. It doesn't go directly to promoter and block the transcription. That doesn't happen. Yet there are more recently people have identified certain silencers. One of the way you can identify it to look for, I don't know if there is a histone code for the same, but since this is a very naive field for the time being, you can look for the binding of certain factors. There are few factors which can function as repressor also. And these are like YY1 factor. YY1 is context dependent. One kind of motif of YY1 leads to activation, another one leads to repression. You have a polycom group of protein. And likewise, there are a few more factors, 4 or 5 factors, which are known to have repressor function, and you can look for their binding sites. Okay, look for their binding site. That's one way you can annotate silencer. The other way that people have looked into is the look for DNA's hypersensitive sites, DHS sites, not occupied by, not bound by known activators. And what you do with that, whether the opening and closing dynamics of these sites are associated with some genes going up and down in the neighborhood. Okay, let's say you have, this is also a method to identify enhancers. Okay, this is common for silencer and enhancers. Let's say you have a enhancer here and you have a silencer here for the same gene, and you have a gene here. So what happens is that look at the DNA's hypersensitive sites. Let's say you perform the DNA-seq assay. Okay, if you do not know these terms, there is a genome technology class in the genome structure course, and maybe I can also cover some of those in this class also. Look at the opening and closing of these elements and look at the transcription of this gene. Okay, this is your mRNA here, with your DNA-seq. Okay. So, and you have to look at across multiple cell types or in time. If you have time course induction of the gene, then you can have this can be your T1, T2, T3. If not, this can be cell type 1, cell type 2, cell type 3. And what you found is that this gene was, when you look at the activation, this was your enhancer, right? This gene was this was not having open chromatin. So the transcription, let's say I plot this is your promoter over here. So this whole region will open up, right? So this was also not opening. It was like that. And it's like that. And when this went up somewhere here, it turns out this has gone up. Consider this as transcription basically, okay? This would not be like that precisely. Let me plot more precise thing. Because opening is more prominent at the promoters and slightly at the terminator also, like that. And then let's say at some cell type or some time point it has gone up here, and when it has really shoot up, then this is also shooting up. So there is a correlation here, right? While if you see other way around, for the repressor, this is silencer here. Okay, now this is silencer, and for silencer, what you observe is that when this was up here, the gene was down, right? When this is low here, or other way around, mixing up. So let us say this was down here. If it is down here, then this will be up, right? And if this is up here, silencer is up, then the transcription will go down, right? So you look at the correlation. So you will have multiple these cell types, maybe let's say 7, 8, or 10 cell types. And then you look at the correlation. How can you calculate the correlation? Correlation coefficient, simple. Correlation where you take this site versus this site, right? And likewise this site versus this side. So look at the correlation of these and these. Then you would know for this enhancer, what is the target promoter? You said this one. For this silencer, what is the target promoter? You said this one, right? So this is one computational inference of identifying silencers and enhancers. Okay. So all these sites which are DNase hypersensitive sites open in one of the cell type, and then people learn that actually none of the transcription factor is binding. Then they learn that in one particular cell type, maybe YY1 was binding there, or there was no transcription activator nearby, so it is likely to be silencer. And then they validate this by this kind of analysis. Right, so this is some introduction to these elements, which can be annotated based on epigenetic modifications and the open chromatin states. In the next class, we will briefly talk about insulator, okay, and then we will look at the principal chromatin types based on several different histone modifications. We have, let's say, 20, 30 different histone modification, right? So we can use, so we have 20 different histone modification for so many sites. Every time you can't look for 20, 30 histone modification and say, well, this is an enhancer, this is promoter, this is transcribed region, this is polycom silence region, this is heterochromatin HP1 associated. You need to compress that information to something called principal chromatin types, that now you say this is active enhancer, this is poised enhancer, this is active promoter, this is poised promoter, this is strong enhancer, this is weak enhancer, this is highly transcribing region, this is weakly transcribing region, this is heterochromatin, this is polycom heterochromatin, this is repetitive region in the genome. So that I just have one annotation, right? So that's called principal chromatin type, but that information is coming from all the different histone modification and by dimensionality reduction of that, okay? So that we don't have to worry about so many histone modification every time. Let us define what the chromatin type is. Chroma stands for color. Okay, so we will have different color for each chromatin type. For example, red color for the promoters, orange for the enhancer, black for the heterochromatin, green for the transcribed region. So that's how it is being annotated. So we will look into that, how people do that in the next class. If you have any question, it's open for the discussion now. Will you record the classes? So this was not being recorded, right? Well, this is not recorded. What I'll give you, I will give you these slides. Okay.
So will you record the future classes? Like,
Well, I can, but I may not post them. I don't guarantee them because I have experience of almost 3-4 semesters online teaching now, and 3.5 in fact, or almost 4. Uh, People don't come to the classes the moment I provide them the recorded lecture, and they don't go through those lectures, and they stack up all the lectures before the exam, and then you have 30-40 hours of lecture to be seen before exam, and nobody looked into that because there is an energy barrier. So I think it's not good for the students themselves. And I can provide you the recorded lecture. I'm giving you the material. I can provide you the recorded lecture. If I'm recording, I will record from the next lecture onward, but I will not publish them. Given a genuine reason, somebody has a genuine reason, then I can consider that. But if you say in one of the polls, somebody said, oh, I had a Makar Sankranti, there was celebration at home, so I could not attend. That's not a genuine reason. Somebody is sick, somebody has internet problem, which can happen in hostels. I can provide the lectures. So I would be generous there, I would be considerate, but by default, no. I'll provide you these slides, whatever I made. This will be available on Google Classroom. All right. Is there any question?
Sir, so there's one question. You were showing some map where there was a difference between H3K4 trimethylation and single methylation. Can you tell me how were you reading that plot? I'm not really sure about it. I haven't seen
Okay.
that first.
This is the distribution plot. Let's say you look at the heights of all the students in a class and plot a distribution. Who all are 4 feet tall, who all are 5 feet tall, who all are 6 feet tall, and so on, right? At different resolution, you make a distribution and then plot it. Like your marks, your and some marks or your total marks. Instructors will make a distribution plot. This is the distribution plot of all the regulatory elements of human genome or mouse genome. Okay. In case you are confused to understand what element, just think of different windows, 200 base pair, 100 base pair windows of the whole genome. That's what I'm plotting. And I'm looking at H3K4 trimethylation at that locus, 100 base pair window divided by monomethylation in that window and next window and all the other windows. And number of those windows with that much mark is on the y-axis. Okay, this is a typical distribution plot. For example, 4 feet tall, how many individuals you plot on y-axis, right? There's a ratio of 0.5 of the ME3 and ME1. How many promoters or genomic sites were having 0.5 ratio is on the y-axis, right? So the height on the y-axis is telling you the number of those sites. Okay, so you don't have a normal Gaussian distribution. There is a bimodal distribution which tells that nature has decided that these two are distinct populations. So this is a natural big gap between enhancer and promoters, which we can utilize to identify in the unknown genome. And when I take a new genome, I'm annotating a new genome, and I have this K4 trimethylation monomethylation, I can identify what all are enhancers and what all are promoters. Is this clear?
Yes, sir.
So, has anybody taught you how do we identify these marks in the whole genome scale? ChIP-seq, did anybody teach you? ChIP, you know, chromatin immunoprecipitation, right? If you do it at genome scale, that's called ChIP-seq. Did Ram teach you this?
We discussed in one class with Shravan sir about how to identify this, but not exactly like the whole detailed
Okay. Okay.
version was not there.
Okay. What I'm going to do is we can briefly introduce chip in the next class, chip-seq in the next class, but not all the other techniques, because then what will I teach in the last section of the course? Okay, wherever needed, if I need to scramble the course structure, don't mind that. Okay, so I can bring the technology part in between. For example, even in this principal chromatin type, I need to talk about some technology also, computational technology. So I will briefly introduce how we identify this at genome scale, different marks. We basically use antibody against monomethylation and trimethylation, separate antibodies, and run a separate chip experiment and do the sequencing. And then you can plot the reads in the genome and then look at how many reads in each bin that you have taken, each window. Okay, 100 base pair window, or 500 base pair window, 1 kb window of whole genomes piled up. Okay, look at the signal for both the things and take the ratio of both the things. Then plot how many such windows were at this ratio level, how many windows were at this ratio level, and plot the distribution. This is like, this could be, let's say, -2 here, because this is log scale, this is +2 here, this is some arbitrary values. It's telling that the monomethylation was high here, trimethylation was very, very low, and trimethylation was low, high here, and monomethylation was low. This is increasing, decreasing, right? And 0 is the one where we generally find cutoff. And if you want to see these, how does the chip-seq data look like? I showed you this aggregation plot over here, right? This is the way this each column is coming from that chip-seq data. Okay, a separate chip-seq for this mark, separate chip-seq for this mark, and around each promoter at this particular promoter, at this particular first row, what was the mark label? Okay, it was low, low, low here, and then it was high here. Next one, it was low, low, low here, and high here, and right. This is the way it is going. Okay, so this is centered around TSS sites, centered around these TSS sites, upstream, downstream, each promoter, upstream, downstream. So generally these sites are coming from the genome, right? This is TSS here, 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 TSS 1, TSS 2, and likewise. So you take this fragment, put it here, take this fragment, put it here, take this fragment, put it here, and make this heat map, right? So these are the bins in which we are looking at the histone modification. What is the histone modification here? What is the histone modification here? What is the histone modification here? And then take them together, take the average of these two, and you get this plot. Right? And if you don't want to take the average, you want to show all of them together, then you need to make heat map. Otherwise, how many curves you will make like that, right? So instead of that, they made this heat map. Right, so I think it's already 6. You might have. Do you have another class at 6? Yes.
No.
Okay. So is this clear? What we are plotting here?
Yes, sir.
This is basically signal at each promoter, right? Each genomic site, you can say, right? In this particular case, we only took promoter elements. You can take the whole genome also at different windows, but right now we have selected all the promoters. Look at the enrichment of S3K4 trimethylation at each promoter and show it as a heat map of color. Blue means low signal, red means high signal, right? And each of these windows that you see, for example, this window is one bin of maybe 500 base pair or 100 base pair, and color here shows how the strength of the signal, how much reads are mapping there. Okay, like this is one bin over here. Okay, so we have all the bins here stacked up like this, and see the signal and plot in as a color from going from blue to light blue to white to pinkish to reddish, right? That's the heat map thing. And for the same promoter, we plotted the corresponding data for S3K4 monomethylation. And then we saw that most of them were low for most of the promoters. The value in the center is low, but there is one population where this is becoming lower and this is becoming higher. What is this population? This is this population, right? This population. All right, so is there any other question or should I stop? So if you have any other question, you can ask again tomorrow, right? So don't hesitate if some plots that I'm showing, which is not clear what is x-axis, y-axis. For example, in this plot, you can understand what is x-axis. These are the genomic coordinates across this locus, right? Y-axis is the signal of DNA-seq, which is telling how much chromatin is open at each of these locations. High means open chromatin, right? Low means closed chromatin and so on. So if any of the plots that I draw casually and I think that those are trivial on my end, may not be trivial at your end, you can stop me anytime in between and ask. I'm stopping this class now.